Cancer cells are dangerous with characteristics of having high rates of cell division and replicating immature cells in your body that function incorrectly with many errors in the DNA code. One way to remedy this is a chemotherapeutic medication, Topotecan, a topoisomerase I inhibitor. Here we see why topoisomerase is targeted. It is one of the enzymes during replication. Replication involves the opening of the double helix and separation of DNA strands, the priming of the template strand, and the production of new DNA segments. Topoisomerase is ahead of the replication fork and unwinds DNA for it to be separated by the DNA helicase. Without the topoisomerase, the helicase would not be able to separate the strands as there is too much tension in the forms of supercoils. There are two types of topoisomerase, but for now we'll be only looking at topoisomerase type 1A, as that is what topotecan is inhibiting. Topoisomerase 1 is characterized of only cleaving one strand, while the topoisomerase 2 cleaves two. Type 1A denotes topoisomerase 1 of having a molecular weight of around 70 kilodaltons and making a phosphatyrosine bond on the 5' end of DNA, while the type 1B is lighter and produces a phosphatyrosine bond on the 3' end during intermediate formation. There are four domains of topoisomerase type 1A with specific roles. Domains 1, 3, and 4, labeled as blue, green, and purple respectively, are made up of alpha helices with dominant presence of basic residues as seen by the primary structure with a pi higher than 7. The basic residues would interact with the DNA's negative sugar phosphate backbone. Domain 4 has a binding groove right here with the turquoise alpha helix, which is not involved in the cleaving but keeps the upstream double-stranded DNA bonded to the whole enzyme. 2 and 3 contain the active site, which is in this black box here, which makes that 5' phosphotyrosine covalent bond with additional polar and basic residues to assist. Domain 2 is mostly a mixed beta plated sheet that acts as a hinge and has a hole above, which both will be discussed later. Looking at the active site closely, this is the general mechanism that cleaves one strand of the DNA through its sugar phosphate backbone. The reaction involves covalent catalysis with the hydroxyl groups of the respective topoisomerase tyrosine and the sugar 5' alcohol as nucleophiles and the phosphate as an electrophile being nucleophilic attacked. We also notice no ATP hydrolysis being coupled as opposed to topoisomerase 2, which requires additional ATP from pathways such as glycolysis and beta oxidation of fatty acids that follows through the electron transfer chain to produce ATP. Instead, torsional strain of DNA itself drives the reaction as the only structural changes are from one monoester bond to another. Here you can see how the domains have their own roles, with the conformational change of the enzyme changing as a covalent bond is made and the other strand is able to pass through the broken strand, lessening the torsion by one turn. Domain 2 is clearly shown to be a hinge for the doors when conformational change is occurring, doors being the domains 1 and 3 holding the active site. The hole by domain 2 creates the space to allow that intact strand to pass through that broken strand, lessening that tension by one turn, that red strand being the intact strand. We have the structure Topotecan and its interaction with topoisomerase cleaving complex. Topotecan mimics DNA base pairs with its heterocyclic rings and binds at the site of DNA cleavage by intercalating between the upstream and downstream base pairs through pi stacking, right here labeled in orange dashed lines, between the bases. There's also consideration of the hydrophobic effect of those rings within the DNA. The other functional groups interact with topoisomerase in the DNA backbone, the hydrogen bonding on its periphery, labeled in blue dashed lines. As for the method group, it takes into consideration when intercalation displaces the downstream DNA. It prevents the religation of the cleaved strand, and the method group assists in preventing the polar 5' alcohol group to nucleophilically attack this yellow topoisomerase tyrosine monoester bond. This keeps the tyrosine cleaved intermediate topoisomerase 1 DNA complex and prevents religation of these single strand breaks. It collapses the replication fork of helicase as there is no template share for one DNA strand. Topotecan acts like an uncompetitive inhibitor in this case, which acts as an already bound complex, reducing Km and Vmax as it interferes with binding of the 5' OH group, keeps the complex from dissociating. One other important mechanism to consider is the cancer's ability to repair these single-strand breaks during replication through single-strand break repairs. The important steps are simplified with part 1 recognizing the gap, polymerase filling in, and ligase connecting the strands together. This presents a problem as cancers are able to proliferate and replicate with apoptosis. However, topotecan can still mimic that of nitrogen bases within that gap, so PARP recognition could be lessened. Finally, as a whole, topotecan can disrupt the central dogma during the replication phase, which The DNA break would be unstable to maintain and lead to cell death. However, if DNA repair mechanisms are in play, cancer cells can go through its normal replication, transcription, and translation steps into synthesizing proteins and tumors. Topotecan is also not a cure because there are many cancer cells that can still be unaffected and many healthy cells being affected with major side effects. Other pathways of the central dogma or certain enzymes can be targeted. We also know that for topoisomerase 2, it can be inhibited through ATPase activity inhibition. All in all, the matter to discuss is around specificity, as Topotecan itself is fairly general, binding to topoisomerase intermediate complex of the major group of BA-DNA. Further studies should be done to target more specific molecules to minimize healthy cell damage and somehow eliminate all cancer cells.